I'm Professor Matt Schmidt, and this is Genetics. We're continuing our discussion of biotechnology, and in this session we're going to focus on an analysis of nucleic acids at the molecular level. And there are a couple of sort of fundamental procedures and ideas that we want to go through in this session. The first of which is a procedure called Southern Blotting. And Southern Blotting allows the detection of certain sequences of interest, for whatever reason you're interested, for an extremely large amount of other DNA sequences, for example, the whole genome. Now, if you think about it, there's something in common with that statement and the screening the library procedure that we talked about if you're going in order in the last session. But we'll see that Southern Blotting is a lot easier depending on your application. It allows you to pick out DNA sequences that you don't want to really do anything with, you just sort of want to get information about them. So the uses include, for example, paternity testing, forensic analysis, creating restriction maps of parts of the genome that aren't even cloned yet. We'll talk more about that in the applications section. The reason it's called a Southern Blot, not because they do it down south, but the process is named for a scientist named Dr. E.M. Southern, who developed this procedure in 1975. Let's take a closer look. So how do you do a Southern Blot? So let's just say you're going to do a paternity test like they do on the Maury Povich show all the time. Not to pick on Maury, but that is a pretty big part of his livelihood. So you obtain a sample of DNA, often genomic DNA, right? If you're getting it from a blood sample or something like that, and you cleave it with restriction enzymes so that many small fragments are created. Again, sort of similar to how you started making a library, right? But now is where it's going to be different. We're going to take those fragments, just the fragments, don't worry about any cloning vectors or anything like that, and perform gel electrophoresis on your sample, which separates the targets in space. I should say the fragments in space. So we're going to talk in a second more about gel electrophoresis, but there's just hundreds of fragments when you cut up the genome like this, and when you put them in an electric field, it spreads them out so that you can have a reasonable sort of a map. It's not really a map, but you'll see in a minute. We denature that DNA that's been spread out, transfer it, you've seen this before too, to a nitrocellulose membrane, which is again basically a fancy piece of paper, and that's the blotting part of the Southern Blot. And then you have a probe for your gene of interest, and you mix that with the blot, and it will bind to complementary sequences only. Then you'll perform autoradiography on the blot to obtain an image, and only the locations where the probe has bound will be visible. So sorry to keep saying this, but there are certain similarities with the procedure we use to make a genomic library. The big difference is you're not taking these samples and saving them and putting them in cells. We're just doing an analysis to see what's there. And that's, if you're doing a paternity test as one example, that's all you'd really care about, right? Now, just as an aside, gel electrophoresis is a very important procedure. Almost every technique where you're doing molecular level things, gel electrophoresis is a part of it. It relies on the fact that DNA is always negatively charged, as it's an acid and there's all those negatively charged phosphate groups around. So if you put it in an electric field, pieces of DNA always will migrate toward the positive electrode, the cathode. Now, if you just put them in water and did that, they'd migrate, but it wouldn't do you any good because you couldn't see them. So you make this gel, and it really is like gelatin. It's a matrix. You put the DNA in that gel and expose it to an electric field. The fragments will start moving, but because of the matrix nature of the gel, what I mean is if you could shrink down and look at the gel, you'd see that there are little holes in it. It's like a porous solid. So the beauty of this is that small fragments can travel relatively quickly through this maze, as it were, whereas larger fragments take a little longer time because they have to snake their way around. But it literally and almost perfectly every time, separates fragments in order of their size from small traveling the farthest to large not traveling so far. And if you use what's often referred to as a ladder, which is a marker containing DNA of known sizes, you can compare your fragments with that and get a good estimate of how big they actually are. Now, you wouldn't need to do this for a Southern Blot, but as long as we're discussing electrophoresis, if you wanted to just look at the gel, literally look at it, ethidium bromide is an agent that we discussed a while ago. It's a mutagen, but the reason it's a mutagen is because it is an intercalating agent which gets stuck, for lack of a better term, in the DNA double helix. It gets stuck in DNA, which means it's not good to touch you, but if you stain a gel with it, it gets stuck in the DNA. And then it has this other property, is that when you shine ultraviolet light on it, it re-radiates the light back in the visible spectrum, and it looks like it's glowing bright orange. So you can literally just either hold a UV light over the gel, and you'll see the DNA band shining. It's pretty cool. All right, but back to the Southern Blot. So the procedure is here. So I'm putting a scenario in this picture where, let's just say it was a paternity test, and there's 3 individuals, number 1, 2, and 3. All of them are possible candidates for being the father. What I've shown is, though, in a particular DNA region of interest, and often it's not a gene, I don't know if these are showing up really brightly, but here are different cuts with the same restriction enzyme, okay? So 3 different people, 3 different patterns, and after you have the cleavage, so in these tubes are going to be floating around fragments of whatever size. Now we're going to keep them separate, separate them on a gel, do gel electrophoresis so that the smaller fragments will travel farther, but basically we're going to be separating out these fragments. Denature the DNA, because we have to do that because the probe is going to need to bind because of the complementary nature. Put it onto the membrane, and then we apply our radioactive probe to the membrane. Literally put it in a plastic bag, let it slosh around, and the probe is only going to bind to the segments that it's looking for, right? So look, guys, if you could see this, you can't see it, but this is genomic DNA, right? There's just tons and tons of fragments all over the place here. You can't see them, and the point is the probe's going to come along, and it's going to say, oh, here's 2 places where it's binding. And that's what you're going to be able to see. And the pattern is likely going to be different for a given individual versus another. All right? So when you do the autoradiography, all you see are the fragments of interest. You don't bother getting to see all those other literally probably hundreds or thousands of fragments that are hanging around there. So you get a pattern. Now individual 3 here has a pattern that's got 3 fragments, and if you look over here, you'll see the reason why. Those are the 3 fragments of 3 different lengths. Now, I sort of did this on purpose, although it sort of ruins the test if it was true. These individuals, in fact, do have different sequences, but the way it cuts, they have 1 large piece and 1 small piece. This guy has a different large piece and small piece, but the pattern sort of looks similar there. This would ruin the test, and it usually doesn't happen because we have identified regions of DNA which are the most useful for doing this type of procedure. But the reason I put it in there is I want you to see that when you do this procedure, it's solely based on the size of the fragment. That's really all you can tell is the probe bound to a fragment of a particular size. Almost always, if you do it right, the pattern would be different from one individual to another. So that's a Southern Blot. Now, there are similar procedures that you can do using RNAs or proteins. So, for example, if you wanted to identify RNA molecules of interest, you could take, here's an example. Say you want to know what RNAs, if a particular RNA is expressed in a certain tissue. You could gather all the RNAs, say, from a kidney in a human being, and